Thank you, everybody, for joining today. So the title is a little broad of exploring progress in narcolepsy research, but it's really going to be focused on treatments. And that's because really where most of the action has been over the last couple of years. And I'm going to start off just with a really quick review of what is narcolepsy. What are the two different types, type 1 and type 2? I'll mention some of the current treatments and how they're good, but how there's room for improvement, and then talk about some things that are in the pipeline, these potential new treatments. And I want to emphasize that word potential because all the stuff I'm going to talk about in that last chunk are drugs that are under development, and things come up, they discover side effects that don't work, that maybe there's some problem with timing or efficacy. And, so not all drugs that are in a pipeline necessarily actually make it into your pharmacy or into the hands of doctors and patients. And so just keep that in mind. So I think, I imagine almost the entire audience is familiar with the basics of narcolepsy, but still just to sort of level set a little bit for anybody who's new to this. Everybody with narcolepsy has some degree of daytime sleepiness. And what we mean by this is that people who are getting plenty of sleep at night still are sleepy during the day. They can fall asleep under sedentary conditions like reading and watching TV, riding in the car, stuff like that. About half of people with narcolepsy have cataplexy, which are these brief episodes of muscle paralysis. They're typically triggered by strong positive emotions, laughing at a joke, seeing a friend you haven't seen in a long time, a pleasant surprise like winning a game. Usually just lasts for, at most a minute or two, and then it recovers. The thing that's so striking, of course, is that when the weakness is severe, people can fall to the ground, and yet they're completely awake. So it's very different from fainting in that regard. There's also plenty of episodes of cataplexy that are just partial, where there's just a little weakness of the head, neck, or voice. We think that cataplexy is the normal paralysis that occurs during rapid eye movement sleep or REM sleep, but it's somehow weirdly intruding into wakefulness. And so your body is paralyzed like that, every time you go into dreaming sleep, and that's normal. But for it to boil up in the midst of wakefulness is weird. And in fact, it kind of goes with these other, we'll call them REM sleep phenomena, where people can wake up and not be able to move. We call that sleep paralysis, or it can occur at the onset of sleep as well. Or they can have dream-like hallucinations that are at the beginning or the end of sleep. Those are called hypnagogic or hypnopompic hallucinations. And so those, we don't usually typically have therapies that target those two things, the sleep paralysis and hallucinations. Sometimes because if you're putting somebody on a cataplexy suppressor med, it helps take care of those, but also because for the most part, they're not as disruptive to people's lives. I mean, they can be bad, and I don't want to minimize them, but usually it's not sort of top of the list in terms of what's really bugging people. Considering that people with narcolepsy are so sleepy during the day, it's really pretty weird that they often have fragmented sleep at night. This occurs in about half of people, and we don't really understand why this should be, but it certainly occurs. It's much more common in narcolepsy type 1. There's other symptoms that I think are sometimes overlooked, and I think anybody who's listening in who's got some narcolepsy appreciates the cognitive aspects of this. People talk about it being inattentive, having brain fog, worrying about their memory. And this has not gotten as much attention in the past, but I think I'll show you a little bit of data of some of the ongoing research. And my hope is that this will become a focus of some of the pharma companies as we move forwards. People with narcolepsy are also found to have a higher than expected prevalence of other sleep disorders like regular old obstructive sleep apnea or central sleep apnea, periodic limb movements, which are kicking movements, REM sleep behavior disorder, where people move during their dreams, or sleepwalking, sleep talking, and things like that. To some degree, this may be a detection bias where people with narcolepsy are going to the sleep lab and then they found to have more than one thing. But also, I think some of this stuff is indeed strangely present, like the REM sleep behavior disorder, the acting out of dreams, that just generally doesn't occur in young people otherwise. Obesity is common. Some of you might know folks who gained a lot of weight at the onset of their narcolepsy. And in adults, body mass index is increased by about 15%. This is compounded sometimes by more night eating, and that can certainly be made worse by some of the meds that we use. Depression's more common. I don't know if that's actually due to the fundamental mechanism of narcolepsy or if it's just a reaction to having a really challenging disorder. And blood pressure tends to be up. And I mentioned this with increasing frequency to all my patients now. It's really good to keep an eye on your blood pressure, talk about it with your doctor, because a lot of the meds that we use can increase blood pressure. And it does seem like narcolepsy itself puts people at increased risk for increased blood pressure. So it's good to be tuned into that and watch it. So narcolepsy with cataplexy is caused by low orexin or hypocretin levels. These are just two names for the same thing. I'm going to use the word orexin through the rest of this talk because it's becoming really the dominant word. But you'll see lots of papers or other things where people call it hypocretin instead. More than 20 years ago now, Emmanuel Mignot and others found that people with narcolepsy with cataplexy have extremely low levels of orexin in their spinal fluid. That's the only way we can measure it. You can't measure it in blood just yet. But in healthy control individuals, you see a range of values that go from the low 200s up to over 600, but the average is in the 300s. In people who have narcolepsy with cataplexy, there's a handful of people with normalish levels, but almost everybody, like over 90%, have these really low levels. In fact, I don't know if you can tell, but there's tons of dots squeezed together at the bottom here. And so if your level is below 110 picograms per mil, that's considered diagnostic of narcolepsy with cataplexy. Now, narcolepsy without cataplexy has normal levels, and we're going to talk about that later. But the relevance of this that's so important is that the orexins are normally important for promoting wakefulness and regulating REM sleep. They act through the two orexin receptors called OX1 and OX2. And if you lose the orexin-producing neurons, you end up with sleepiness and cataplexy because those circuits that normally wake you up and control REM sleep are just not working well. This is what it looks like if you look at slices of brain from people who have died, who had narcolepsy for most of their life. These are, typically we look at this when somebody dies of other causes when they're 75 or 80 years old. And in a healthy control individual, meaning somebody without narcolepsy, we see even on one section, hundreds of orexin-producing neurons sitting in the hypothalamus. And in somebody who has narcolepsy type 1, there's typically about 5 or 10% of the normal number of neurons. We're pretty sure that this is due to an immune system attack on the orexin neurons. And it's probably through a process that's called molecular mimicry. So let me just talk you through that. So I have to say, let me just back up for a second. This is a theory. We don't have direct evidence, but there's growing evidence that this is the case. Since the 1980s, it's been known that people with narcolepsy often carry a gene. And some of you might have been tested for this. It's called HLA DQB1 star 0602. That molecule is a type of what's called MHC class 2 molecule, this little blue squiggly thing here. When you have an infection, pathogens, meaning bugs, bacteria, viruses, things like that, get gobbled up by what are called antigen-presenting cells. These are sort of like amoebas in your immune system that eat up bacteria and viruses, chop them up into little pieces, and then they present those little pieces to T cells, which are immune cells, using those MHC class 2 molecules. So right here, this is the gene that's relevant for narcolepsy. And so something about having that gene means that these amoeba-like cells are showing the immune system things a little differently than in other people. And so what that does is it activates their CD4 and CD8 T cells, which are immune cells. They make chemicals that help kill bacteria called cytokines. And so they do their nice job of killing off the bacteria and viruses. But we think the hypothesis is that what goes wrong in narcolepsy is that you get these jazzed up T cells that are great for fighting strep or flu or whatever it might be, but they cross-react with something on the orexin neurons. And it might even be the orexin proteins themselves or their precursor. The precursor is called preproorexin. But so basically what you got is you have these immune cells that are all well-intentioned. They think they're killing bacteria and viruses, but they accidentally kill off the orexin neurons. That's the general theory. The evidence for that is that people with narcolepsy often have T cells in their blood that are activated by this preproorexin peptide. And so this is the going hypothesis. The trouble, of course, you'd say, oh, well, when somebody gets sick, just keep the T cells from killing the orexin neurons. That would be great. But the trouble is most people come into clinic and get diagnosed many months or typically many years after the orexin neurons are dead and the symptoms are well established. And so we often end up diagnosing it way too late, and the cells are already very, very damaged. So that being the case, a lot of the focus these days is on trying to make up for the loss of the orexin neurons rather than trying to save them in the first place. So I've mentioned these two terms, and I just want to tell you exactly what they really mean. We recognize two types of narcolepsy type 1 and type 2, NT1 and NT2. NT1 is the classic form. This is the one that's associated with low orexin levels. And to have a diagnosis of narcolepsy type 1, you have to have sleepiness and then a history of cataplexy plus a positive multiple sleep latency test. And what that is the MSLT is a test where you're asked to fall asleep 4 or 5 times across the day. And if you fall asleep, you can see it at the bottom here, on average in less than 8 minutes. And if at least 2 of those naps contain REM sleep, that's considered a positive multiple sleep latency test. An even better and more concrete way to measure, to diagnose this is to do a spinal tap and measure orexin levels in spinal fluid. It's just not done a whole lot in the US. And to be honest, it's somewhat, in many ways, more straightforward than getting a multiple sleep latency test. It's a nice sort of yes or no answer. Either your levels are low or they're not. But, I can understand a lot of people are kind of averse to having a spinal tap. But I find it, we don't do it very often, but it actually is very helpful when the diagnosis is somewhat ambiguous. Okay. And so I'm just going to finish with this slide, then I'm going to go to the 2 questions that have popped up. So narcolepsy type 2 includes sleepiness. There can be hypnagogic hallucinations. There can be sleep paralysis. But the diagnosis is basically made by that positive MSLT. And it's a little bit of a squishy diagnosis because the MSLT can be positive if somebody's not been getting enough sleep, or if somebody's recently stopped antidepressant meds, that can make REM sleep pop up during the daytime. And so there's things that can mess it up. And so when somebody has narcolepsy type 1, I ultimately am usually pretty confident about do they have it or not. With narcolepsy type 2, it's harder to have that certainty. And of course, there's no point in measuring orexin levels because they're going to be normal. It's not terribly helpful diagnostically. So Chris is asking, would you call narcolepsy type 1 a neurodegenerative condition? Wait, actually, he wrote NT1, NT2. So first, we have no evidence that NT2 involves any cell loss. But for NT1, where the orexin neurons are gone, you could call it neurodegenerative, but I think that kind of is a scary sounding term. You're not putting it into the same category as Alzheimer's and Parkinson's. I think it's probably best to think of it more as an autoimmune disorder, where you have the immune system going bonkers and killing off these cells accidentally. Liam is asking, how likely is it that in NT2, the immune system is targeting only the molecules of orexin? And maybe that's an interesting idea. And it's a little bit hard to test, but I'm not aware of any data for or against this. But I think what Liam is asking is maybe those cells are still making orexin, but it's being attacked by the immune system. So it's not be able to do its job. That's an interesting idea. And it's something, honestly, it sort of feels like we can pat ourselves on the back for having figured out narcolepsy type 1 over the last 20 years. But what's super embarrassing? Half of the people in the country have narcolepsy type 2, and we don't know anything about it. It's really a huge gap. So Chris is asking, what do you recommend to decrease the time to diagnosis of narcolepsy? Oh, boy, really the best thing? Increase awareness amongst pediatricians, school nurses, the general public. Partially, it's on those providers that people see when kid develops narcolepsy at age 14. If a school nurse knew what narcolepsy was and a kid goes in saying I'm napping all the time in classes now, maybe we could get an early diagnosis. It's a little bit on the patients too, and I don't mean this to sound sort of guilty in any way, but I just I think when people are sleepy, sometimes they just kind of sweep it under the rug and they muddle through for months. But certainly, I think many of you probably can remember very clearly when it started, and I think some people get to the doc fast, others don't. Amber is asking, is narcolepsy autoimmune or neurological? And in Amber, it's both, right? It's an autoimmune process. So the immune system is going crazy, but it's killing part of the nervous system. And so in that sense, it's a neurological disorder. It's mostly been studied by neurologists and sleep specialists. And I wish there was more immunologists who actually cared about it, because if there was a dozen labs in the country working on the immunology of narcolepsy, that would be great. That would produce some real progress. Kira, let's save your question on natural remedies for a little bit. If we could just leave that up on the list. Beth is asking, if somebody's diagnosed with narcolepsy type 2, is it possible that it could change to type 1? And the answer is yes. So here's how that goes. I'm going to flip back a few slides. Here, you can look at spinal fluid levels of orexin and look at the right-hand group there, the narcolepsy without cataplexy. Do you see how it's not a huge number of people here? There's only like 21 subjects or 20 subjects studied in this. But there's a few of them that have low levels. By definition, they did not report cataplexy in this study, but their levels are kind of low. And so if you do a large enough sample of people with narcolepsy without cataplexy, about 5 or 10% of them actually do have lowish levels. And of those, over the next years, they start to have cataplexy. It may be that nothing came up that really triggered it, or maybe their cell loss got a little bit worse over that time, but it's an interesting question. And it sort of points to the fact that there's a little bit of a fuzzy boundary between type 1 and type 2. Okay. What research is being done for narcolepsy type 2, asks Susan. I'm afraid to say, beyond the clinical trials, I'm not aware of a whole lot of either basic research or clinical trial stuff that's really trying to unravel that. And part of the challenge is we don't really have good biomarkers. You know, with narcolepsy type 1, it's like, oh, orexin deficiency, let's study that. And that's really what my lab does. But we need better handles. You know, we need to find something concretely and consistently wrong in narcolepsy type 2 to really get that toehold that would allow for some good research progress. Matt's asking, do you believe orexin loss is due to destruction of, I think that means of orexin producing cells or epigenetic silencing? Wow, that's a very sophisticated question. I could go on this topic for a while, Matt, but basically, I'm not really convinced of the epigenetic silencing. I think it's a clever idea. I think I would like to believe it's true, but the one paper that was suggesting that has some, in my mind, pretty substantial problems. And I think there's substantially more evidence on the other side that the cells are truly dead and gone. And in support of the cells really being gone, I'll just say that we and other people have found other markers for the orexin-producing neurons besides orexin itself. There's other genes that are expressed in those cells. And when we look at those in the brains of people who have died, who have narcolepsy, the other markers are gone too. And so if you're seeing multiple, if multiple markers are gone, it really suggests that the cells are truly gone. Okay, Maurice. I'm wondering if I can answer it. It almost sounds like a personal question. I'm 65 years old. I had my first narcolepsy at 5, and at 7, my first cataplexy. Is there progress to minimize the impact of cataplexy between that gap of time? Yeah, okay, good point. All right, so what Maurice is getting at here is that sleepiness almost always occurs first, and the cataplexy follows months, well, sometimes weeks, but typically a couple months, and sometimes even a couple years later, as in Maurice's case. That, I think, is a very important point, because remember how I was saying it's like, people come to clinic too late, we can't do anything. If narcolepsy were really identified early, before the cataplexy develops, my guess is there's probably some orexin neurons worth saving, and that you didn't have cataplexy for 2 years because maybe you had only an 80% loss of the orexin neurons, and then it got worse over the next year or two, and it went down to 90, 95%, something like that. So I do think that's a potential window for intervening. One problem is that people have done very tiny trials of immune system modulating drugs, and they have not really found clear evidence that we can stop that progress, but I still think it's worth trying. And so I would like to see more of that kind of thing. Oh, Iris is asking, are both types incurable? Depends on what you mean by incurable. Can the symptoms be made better? Absolutely. The current meds are generally pretty good. The future meds, I would like to believe, are going to be even better. And so in that sense, we can make a big difference in how people feel. Can we make the orexin neurons come back? Probably not. Maybe someday in the future, but I'm not holding my breath for that. That's a very complicated thing. For narcolepsy type 2, so I'm talking about narcolepsy type 1. For narcolepsy type 2, it's anybody's guess, because if we need to understand the underlying mechanism first to find a cure. Yeah, and so I see Liam's question here about the antigen-specific suppression. So we've been in touch, and I know what you're talking about. So I think it's like, sure, I would like to see people move ahead with any kinds of immunomodulatory therapies possible. Solomon, this is going to be the last question for now, because I need to get to some of the other slides. But how can the terminology used towards cataplexy be changed to better articulate the actual human experience? Cataplexy gets such little focus. There's a massive disconnect in the comprehending of cataplexy for what it is, the human experience. For instance, muscle weakness is flawed terminology in reference to the experience. It's part of excessive sleepiness, sleep attacks. Yeah, it's an interesting question. I think this would be part of a much bigger conversation, Solomon, because I get it in the sense that when people have cataplexy, they just want to crumble, and in that sense, it has that sense of heaviness, like as you can imagine with, when your sleepiness is really bad. But people can have cataplexy when they're feeling really alert too, and in that sense, I think it's best to look at these as kind of different dimensions. Cataplexy affecting muscle power, sleepiness affecting overall alertness level. Laura, I think you're asking a question about LUMRYZ, and so we're going to save that for a little bit later, okay? All right, so, and Kira's question I'm going to get to as well. Thanks for all the questions, you guys. I got to make some progress here. Hold on. All right. I've kind of touched on this one already about narcolepsy type 2. And just to show you how murky it is, because I was talking about a lack of biomarkers, there's one postmortem study where they looked at the brains of people with narcolepsy type 2. And they had intermediate levels of cell loss of the orexin neurons. One person had 33, the other one had 85% loss. So it's possible that narcolepsy type 2 is on this spectrum, that the cell loss is not as severe as narcolepsy type 1. But with only 2 patients, it's hard to take that to the bank. We need to get more postmortem tissue. And as I mentioned before, about 20% of narcolepsy type 2 patients actually have low orexin levels, and about 13% are in that intermediate range. And so, I'm open to this idea that it's kind of the same thing. It's just not nearly as bad as NT1 in terms of severity of cell loss. And I mentioned earlier that among people who have narcolepsy type 2 and have lowish orexin levels, about 1/3 of them actually do go on to get cataplexy. Maybe, as one of the questions was touching on, maybe people with narcolepsy type 2 make orexin peptides, but then there's a downstream problem, like the receptors are messed up or there's something blocking the receptors. And I'll show you some evidence that suggests that there's probably not a problem with the receptors. But it could also just have nothing to do with orexin at all. It could be that it's just similar looking, but completely different cause, and we just haven't figured it out yet. Okay, so turning to treatment, and that's where the rest of our talk is going to go. One of the questions was, what about natural remedies? And I really think, I'm a little embarrassed to say that when patients come to clinic, we often spend a lot of time talking about meds and side effects and doses and and I really feel like we don't talk as much about the non-pharmacologic stuff. And I wanted to at least touch on that briefly here. I think with these brown bag lectures, this would be a really great topic of just behavioral, what can you do that's not meds? And I think I've just touched on a few of the high points. Like, I think it's really important to understand what's an optimal amount of sleep for you. Some people are long sleepers, and maybe you just feel a lot better with 9 hours of sleep rather than 7.5 hours of sleep. If you're kind of on that spectrum towards idiopathic hypersomnia, do you feel any better with longer sleep? Are naps helpful? And for most people, the answer is yes. Many people with narcolepsy love naps because they feel much more alert afterwards. And it's really important to use those strategically because it can really help minimize the need for meds. What helps you wake up, right? What makes you more alert? If you're getting drowsy at work, standing up, walking around, working with other people, working with bright lights, standing desk, whatever it is, there's all kinds of tricks that can be helpful. But also thinking about what makes you sleepy. Everybody with narcolepsy really suffers with sleep deprivation. And it's just gonna, now you got two things making you sleepy. And so make sure you're getting plenty of sleep. Try not to, if you're traveling across time zones, think about optimizing your sleep that way. Avoid sedating meds. Think of whether, consider whether heavy meals make a difference. I've certainly heard from plenty of patients that a big carbo-heavy meal can make them sleepy. And then talk to other people through groups like Wake Up Narcolepsy, Narcolepsy Network, Hypersomnia Foundation, Project Sleep. And importantly, get support from your family and friends, right? I'm preaching to the choir here, but you guys, I think I have met so many people who are just kind of a wreck. And they say, I've never met anybody with narcolepsy before. I feel like the only one struggling with this. And so, going to these groups and getting some understanding from your close family and friends is really super helpful. Okay, I want to quickly define a couple of the clinical tests. I'm going to get to the questions that are accumulating over there and when we start talking about meds. I think if any of you have narcolepsy, you've almost certainly had a multiple sleep latency test. And this is where in the sleep lab, you're asked to try and fall asleep. And we do this every 2 hours, 5 times across the day. And what we're looking for is how quickly do you fall asleep and do you have REM sleep? Those REM sleep containing naps are called sleep onset REM periods, or SOREMs. And I mentioned before, if you fall asleep in less than 8 minutes on average and you have 2 naps with REM sleep, that's supportive of narcolepsy. But that test can be falsely positive and falsely negative. And one of the things that can foul it up is not getting enough sleep on the preceding night in the sleep lab. The rule is you're supposed to have at least 6 hours of sleep. But it's a terrible place to sleep. The mattresses are hard, it's weird, all the wires and things like that. And a lot of people don't sleep so great. And so we actually just we're trying a new protocol in our lab where if somebody doesn't get 6 hours during the normal period, we'll let them sleep late and just drop one of the naps because we want to make sure that they're reasonably sleep satiated. Actually, there's a paper from Australia just recently that did that, letting people sleep ad lib overnight in the sleep lab. And they found that people stayed awake longer on the multiple sleep latency test, and there were fewer episodes of REM sleep. And that really tells you that those lab conditions can bias the test. Your doctor should be telling you to get off your antidepressants for 3 weeks beforehand, no wake promoting meds for a week, and you need to be getting what is for you an adequate amount of sleep, whether that's 8 hours or more on the week or 2 running into the tests. Okay, in the drug trial studies that I'm going to show you, they use a different kind of test, and they use this thing called the maintenance of wakefulness test because it's really stringent and really challenging. And here, the instructions are just the opposite. You need to try and stay awake for 40 minutes, 4 times across the day while sitting in a comfortable chair in a dimly lit room doing nothing. It's super boring, and even healthy people fall asleep on this test. And what we're measuring is the average time to fall asleep. We do use it in the sleep clinic occasionally, like if somebody's trying to get back their license or we want to show that their meds or their CPAP or whatever is working properly. But it's very commonly used in the trials. And at the bottom, you can see the normal values. For somebody with narcolepsy type 1 who's not on any treatment, even though they're instructed to try and stay awake, they often fall asleep in 3 to 5 minutes. On meds, if you look at the stuff that's been published over the last 20 years, people stay awake for about 5 to 13 minutes thereabouts. If you look at the general population without narcolepsy, they can typically stay awake for around 30 to 35 minutes. Depends on the study. Sometimes it's in the 20s. I'm not going to talk a lot about idiopathic hypersomnia, but it's kind of a cousin of narcolepsy. They also have trouble with daytime sleepiness, but they also often have a really hard time waking up in the morning. And many of them have a tendency towards long nighttime sleep periods. And so the multiple sleep latency test similarly shows an average sleep latency less than 8 minutes, but they don't have more than 1 sleep onset REM period. Another way of diagnosing it is to see if somebody's got long sleep night after night. If they can sleep more than 11 hours night after night, that's very indicative of idiopathic hypersomnia. In contrast to narcolepsy, people with IH tend to have very long naps and they don't feel rested afterwards. Okay, so here's a table that summarizes the differences between narcolepsy type 1, type 2, and IH. And all of them share daytime sleepiness. That's a feature of all. Cataplexy is only in narcolepsy type 1. Low orexin levels are only in narcolepsy type 2, except for those kind of rare patients we talked about for NT2. 2 sleep onset REM periods are seen in NT1 and NT2, but not IH. That's how the diagnosis is made. Long sleep is common in IH. Sleep inertia, meaning a hard time waking up in the morning, is very common in IH. You see it sometimes in NT2, but most people with narcolepsy actually pop awake in the morning pretty decently. And then this gene that I mentioned, the HLA DQB10602, it's found in over 90% of people with narcolepsy type 1. It's found in about 50% of people with narcolepsy type 2. And it's found in like 12 to 25% of people with IH. Okay, so to the meds. So this is the current meds that we're using for sleepiness. And I've listed here sort of the range of doses, the kind of side effects, and just sort of a rough approximation of cost. And so, since the 1940s, we've used various stimulant medicines, methylphenidate, dextroamphetamine, that kind of stuff. They work. They have a lot of side effects. They're cheap. I don't really want to spend a lot of time on these meds because I think most of you have probably had some experience with them. Modafinil and armodafinil came out about 25 years ago, and they are, I would say, preferable to the stimulants because they're pretty gentle. Side effects are fairly uncommon. They're not terribly expensive. Nice, decently long duration of action. Oxybates are those weird medicines, the liquid typically that you take at bedtime and then often midway through the night. We don't know how they work, but they do help improve alertness during the day and they help suppress cataplexy. And so the 3 brand names that are Xyrem, Xywav, and Lumryz. Super complicated meds because the side effects are fairly common. They're incredibly expensive, and it really is a big conversation before starting somebody on any of the oxybates. Laura was asking, have there been studies on the effectiveness of Lumryz for narcolepsy type 2? And Laura, my recollection is that when Avadel did their studies of Lumryz, they just lumped NT1 and NT2 together. And I have to say, I can't off the top of my head pull out whether there was much of a distinction between them in the results. I think that they showed equal effectiveness in NT1 and NT2. I'm going to save Kira's question. Okay, I think I'm gonna try to get to some of these other ones later because I just don't want to keep people past the top of the hour. So, all right, so that's our current meds for sleepiness. Oh, I didn't mention the bottom ones. Oh, solriamfetol, which is Sunosi, or pitolisant, which is Wakix. These have just come out in the last 3 years or so, and generally pretty good medicines. Sunosi and Wakix both have relatively few side effects. Sunosi is kind of medium expensive. Wakix is quite expensive. They're both, I would say Sunosi actually, I would say it's very good at improving alertness during the day. Wakix, I would say, is similar to modafinil or modafinil. It's nice, solid medicine, well tolerated. And the thing that's kind of funny with it is some people say it doesn't really do much for them. Other people love it, or it really seems to make a big difference. And so it's one of those things where I just, you got to, if you're not doing well, it's probably just worth trying it and see if it works for you. Okay, I want to talk about oxybates a little bit more because I think this is maybe a source of some confusion. 20 years ago, the original oxybate came out. So they're called oxybates because that's the chemical structure. So sodium oxybate is Xyrem, and the generic that's out there is also sodium oxybate. It's the exact same thing. And so it's a mixture of literally sodium and this oxybate, which is the active drug. And 9 grams of Xyrem, or the high salt oxybate, is 1.64 grams of sodium. And your total dietary sodium is really supposed to be, depending on the guidelines, less than 1.5 or 2.3 grams per day. So if you're taking the high dose of Xyrem, 9 grams, you're basically doubling your sodium intake. And that's not necessarily bad. Some people are fine with that. But you might remember earlier I said that people with narcolepsy can have a tendency towards elevated blood pressure. And so I think it's worth keeping an eye on blood pressure if you're taking a high salt oxybate. For the last, I don't know, 10 years or so, there's been this low salt alternative where they wanted to get away from that high sodium content. And so they made Xywav, where they replaced the sodium with calcium, potassium, and magnesium. And it has about 1/10 as much sodium in it. And so, from a blood pressure perspective, I think there's a pretty good, if you have high blood pressure and you're on Xyrem, you probably should switch to Xywav. I'll put it that way. If the lower sodium helps with your blood pressure. A year and a half, 2 years ago, Lumryz came out. This is the same chemical, but it's packaged in a way that instead of having to take a dose at bedtime and midway through the night with the conventional oxybates, you just take it at bedtime. It comes as a powder, whereas the others come as a liquid. So you mix it into orange juice or something like that. And it really seems to be very similar. So if somebody's on 9 grams of Xyrem, they can switch to 9 grams of Lumryz or vice versa. And so I don't think there's any more or less safety concerns. But it's just a little different process. With the liquids, you measure them out. With the Lumryz, it comes in fixed packets of 4.5, 6, 7.5, or 9 grams. So the advantages of the conventional oxybates are that you can play around with the dosing. And so, for doctors who are experienced with them, you can kind of nuance it a little bit. You can do a big dose and a little dose. You can do 3 doses, and we can sort of adjust the dosing to suit the individual's needs. However, having said that, I've had some patients who just have a really hard time consistently taking that second dose. And if they don't take the second dose, then they have much less benefit during the day. And so that's one argument for why the once nightly is good. The once nightly is also just sort of, I say, conceptually more convenient. Though the downside is that you can't sort of nuance the dosing like you can with the liquid ones. Okay, for cataplexy, these are the meds that we use. And up top, you can see venlafaxine and fluoxetine. These are, and clomipramine. Those are all antidepressants. And we're not giving them because this hasn't much to do with depression. It's just that neurochemically, there's some similarities. And so venlafaxine and fluoxetine increase norepinephrine and serotonin. And as it turns out, that suppresses REM sleep. And that means you're suppressing cataplexy along with it. So we can often get away with very low doses of those antidepressants. And they're cheap and they're often very effective. Oxybates are also great at suppressing cataplexy, as is pitolisant, except those last 2 meds are pretty expensive. Okay, so here's more or less where we are in terms of how well we're doing. This is a study by Kieran Maskey from a few years ago. And I have to say, solriamfetol and pitolisant were not available at that time. But this is basically people with narcolepsy type 1 and type 2 who were on meds already. And 75% of people with NT1 said their sleepiness was still a problem, even on meds. 88% of NT2 said that. 47, let's just call it 50% of both groups said that they were having cognitive problems with difficulty thinking, paying attention, and so forth. And so what you can see is that, while the meds are helpful, they're not perfect by any means, and there's still a lot of room for improvement. And, fatigue and disrupted sleep continue to be problems for people. And so this sort of sets the stage for why should pharma companies bother developing new medicines? Because people need better stuff than what we've currently got. So here's a list of some of the drugs that are under development. And I first want to point out, this is a really long list. It's kind of crazy. 5 years ago, prior to the last few years, at any time, there was rarely more than 1 drug in development for treating narcolepsy. And now look at this, there's tons. And that's great for anybody who's got this. So reboxetine is the one that's closest to the finish line. This is a medicine that's really being developed primarily to suppress cataplexy, but it also helps with wakefulness. And we'll talk about that in a little bit. Mazendal is being mostly developed in Europe, and I honestly, it seems to be in the phase 3, but I'm not quite sure if it's even going to what's going to happen with it. It's been available in Europe for years, and to be honest, my narcolepsy research colleagues there have not been super impressed with it. So I don't. I guess what I would say is it might be helpful for some patients, but we need to see. The serdexmethylphenidate is essentially a long-acting methylphenidate. I think it's sort of a delayed release, where it's kind of like, well, anyway, you can take it at bedtime, and it then kicks in the day. That's in let me talk about drug phases. So when a drug is first developed, the first thing is to test it in animals and test it on the bench, and that's called preclinical testing. The first human testing is called phase 1, and that's when you try the drug in small numbers of either healthy individuals or people with narcolepsy. Phase 2 is when you go into a bigger group, like maybe 40 or 60 people with narcolepsy, and really convince yourselves that it's going to work. And then phase 3 are the big studies, where typically a couple hundred people take the drugs, and you get really good, deep information on whether it's going to truly be effective and is it truly safe. So what you can see on the right hand is that these are all in different development phases. And I highlighted in the pink colors this cluster of orexin agonists, and that's where I'm going to put most of my focus, because these are really unique. The other mechanisms that are up on this list are kind of in some ways similar to some of the current medicines, but the agonists are something quite novel, and I think there's quite a lot of excitement around that. So, but before we get to those, let me just talk about reboxetine, or the sort of code name is AXS-12, and that's been developed to decrease cataplexy. And so they've done a phase 3 study of a decent number of people with narcolepsy type 1, and to get into this study, you had to have at least 6 episodes a week of cataplexy, so pretty decent amount. And on the right, you can see week by week how people did, whether if they got placebo or the reboxetine. And it's pretty clear that with the reboxetine, there's a really nice suppression of cataplexy. And on the bottom left, you can see the percent of people who actually had no cataplexy at all in the week. And at baseline, they had to have at least 6 episodes a week. And then at week 1 and at week 2, 3, you're getting up to this range where, in any given week, 20, 30% of people didn't even have any cataplexy, whereas the placebo groups, only maybe 5 or 10% were in that category. So this was not the main endpoint of this study, but reboxetine also did reduce sleepiness. It increases norepinephrine in the brain, which helps wake you up, and that could be part of it. But on the y-axis here, this is called the subjective clinical global impression scale for sleepiness. And so it's really patients just saying subjectively that they feel less sleepy. And you can see there's a nice difference between the placebo and the drug groups. And the percentage of people who were taking naps went down in the drug-treated group in pink. Okay, so I'm going to take Kira's question here. She was asking, is there a possibility of lab-made orexin neurons that can reverse narcolepsy? And Kira, that actually has been tested in a couple of animal studies where they took narcoleptic animals and put new orexin neurons into the brain, and they did get better. The reason that has not been pursued in humans is because working with cells like that, it's really a challenge in so many different ways. One is, well, if the immune system killed off the last orexin neurons, how are you going to keep them from doing that again? Second, how can you get those orexin neurons to truly grow and survive and connect to the right places so that they're behaving normally? And because it's such a technical challenge, and because there's that worry about immune responses, it's not been a very active area in the last few years. Maybe we'll come back to that if people get really great at playing around with stem cells in the future. But right now, I'd say it's not something that we're waiting for. And that also applies to giving orexin peptides as a treatment too. You might have seen a few papers on this, and I have the same concern. If immune cells are attacking the precursor protein, who's to say that giving somebody a bunch of orexin peptide isn't going to just stir up the same immune response? To compound it further, if you were to basically eat a bunch of Orexin, it would get chopped up in your GI system. And even if it got into your bloodstream, it gets broken down there too. And so Emmanuel Mignot injected Orexin into the bloodstream of some dogs, and only 1% of it got into the brain. And so it's just really hard with the natural Orexin peptides to get them into the brain in the right way. And that's why people are trying to make drugs called Orexin agonists that mimic the effects of those. And so the goal here is you make something that's not an Orexin peptide, meaning a protein, but it's a small chemical that binds to the receptors and does the same thing as the native Orexin peptides. And the fun part of this is that it's really targeting at fundamental Orexin deficiency. That's the core element of narcolepsy type 1. And so the expectation is that the efficacy should be quite good with these. Because there's similar symptoms, the hope is that it may also help narcolepsy type 2 and IH. And it may allow us to, if you're only trying to get somebody to a normal level of orexin signaling, maybe you can do that with a fairly minimal amount of side effects. But there may also be surprises. I mean, there's, with any new class of drugs, there's always going to be unexpected stuff. So I'm going to show you some examples of this. This is a paper from New England Journal of Medicine about a year and a half ago. And this was a drug called TAK-994, and some of you might have actually been involved in these clinical trials. But what you can see here, so after 8 weeks of treatment, people who got placebo were falling asleep on the maintenance of wakefulness test in only 4 minutes. The folks who got the low dose of this drug, 994, stayed awake for 31 minutes. And with the higher doses, they were staying awake for 35 or really 40 minutes. I mean, that's amazing. They're staying awake for the entire test. The Epworth sleepiness score, which is that subjective feeling of sleepiness, starts off in the moderate range at about 14, and it went down to 5, 3, or 1 with the higher doses. And the cataplexy went from 10 episodes a week down to about 1. And so that's amazing. And everybody was so excited. And then it was so heartbreaking because this drug was associated with some liver inflammation. And so Takeda, the company making it, had to stop its development. It was a big disappointment. But it tells you that's what I was getting at earlier. There's going to be bumps on the road with anything new like this. So Takeda now has another drug called TAK-861. Oh, I should mention that all of the orexin drugs that I'm going to be talking about are binding only to the orexin receptor 2. There's 2 orexin receptors, OX1 and OX2. But OX2 does most of the heavy lifting for sleepiness and cataplexy. And so that's what the drugs are being designed to accomplish. So this is some data from a reasonably good-sized study, from a phase 2 study of TAK-861. And what you can see here is that at baseline, on no treatment, people with narcolepsy type 1 were falling asleep in 4 to 6 minutes on the maintenance of wakefulness test. After 8 weeks of treatment, the placebo group in gray was basically the same, 4.7 minutes. But with the varying doses of TAK-861, you were seeing people staying awake for 16, 20, 30 minutes, something like that, which is really very respectable. Earlier I was talking about what's normal on the maintenance of wakefulness test, and it's kind of debatable. Some people say better than 30, some say better than 20. And personally, 20 minutes is still a giant improvement over the kind of effects we're getting now. And so these 2 medium doses, the yellow one, which is 2 milligrams in the morning and 2 milligrams 3 hours later, or the blue one, which is 2 milligrams in the morning, 5 milligrams 3 hours later. Those were good. 27 minutes, 30 minutes on this test. I'm going to take the last questions at the end, but we'll see how many we can get through. This is showing Epworth sleepiness score. So this, again, is subjective sleepiness. At baseline, on no meds, people were really sleepy. Epworth scores of 18 to 19. With treatment, the Epworth scores were down in the single digits. This is great. This is so important. 4 minutes, 5 minutes, that's normal, really normal. And so that's super encouraging because people felt clearly more alert on this med, subjectively. So this is something that y'all may not have seen before. This is a poster that was shown at the European Sleep Meeting last fall. And here, some Takeda researchers looked at the psychomotor vigilance test. This is a test where you're looking at a computer screen, a little dot or a light is going to flash on the screen, and you have to push the mouse button or a key as fast as you can. And you have to do this over the course of, I forgot if this was a 6 or a 10-minute test, but it's boring. You're just looking at the screen, waiting for something to pop up. And so at baseline, without any meds, about 15 to 20 times across the test, people were failing to respond. These are called lapses. And so the little light flashes and the person doesn't even click the button because they're just not as alert and focused and paying attention as they should be. But with treatment, with the various doses, what you can see is that the number of lapses went down to 5 or 10, which is a big improvement. Normal is probably maybe having 3 lapses on the test. So it's not quite normal, but it's still better. And what I really like about this is simply the fact that it's starting to be measured now. Actually, I should point this out. Look at the error bars on this. There's a tremendous amount of variability too. And so in the future, it'll be nice to talk about how much each individual improves, because some people are going to be better or worse on the test. But I'm glad to see that pharma companies are paying attention to cognition as an endpoint. Okay, this is what's called the long-term extension study. So the proper phase 2 study was only 2 months long. And what you can see here is on the maintenance of wakefulness test, people got a lot better compared to baseline. But what's nice to see is that they then receive the drug for the next 6+ months. And at month 8.5, their responses are still just as good as they were at month 2. And similarly, the amount of cataplexy stayed down. So that's really great because, one of the concerns with anything new is like, well, is it going to hold up over time? And so this is nice evidence that you can take this medicine for 8 months and do just as well as you did as when you started. Side effects with these orexin agonists are kind of the same across all of them. And they're kind of weird because nobody would have predicted this, but frequent urination is one of the top things. Some people say it feels like drinking too much coffee and that people end up just having to get to the bathroom faster. And sometimes they really have a sense of urgency where they got to pee right now. Insomnia is pretty common when people first start these meds, but it tends to sort of fade over the first week or so. And it's generally not a long-lasting thing. In general, the side effects with these meds have been quite minimal. And I would say probably the best evidence for that is that at the end of this 2-month period, people were given the option to continue on and get free drug for the next 6 months or so. And I've forgotten the numbers, but like 90 to 95% of people opted to continue on with the study drug and rather than go back to their regular meds. And I think that's a pretty strong argument that they felt that the efficacy was so much better that they were willing to put up with whatever side effects they might have been having. Okay, this is a new drug from Alkermes. They're in phase 1, so they're doing small studies. And so here, this is just 10 subjects. Okay, much smaller. I didn't really point it out, but I forgot. There's like 40 subjects or something in the phase 2 studies I just showed. But here, the Alkermes drug looks just as good. And the doses are low, 1, 3, 8 milligrams, and you get these really terrific improvements on the maintenance of wakefulness test, where they're staying awake for almost the entire duration of the test. Side effects are very similar: insomnia, urination, etc. This is a study in people with narcolepsy type 2. It's a very small study, only 9 people, but I want to point out that the improvements look just as good. You have to give higher doses than you would for narcolepsy type 1, but you can get people staying awake on the MWT test for a long time, right up into the, into the 30-minute range. And that's terrific. Side effects, again, are very similar. Sorry, I'm going fast now just because I don't want people to have to stay late. Alkermes and Takeda actually have also done studies in people with idiopathic hypersomnia. And here you can similarly see these nice improvements in ability to stay awake on the maintenance of wakefulness test. So I'm hopeful for people with NT2 and IH that some of these meds are going to be beneficial, just as it is in NT1. I'm going to skip these last couple slides and just go to the conclusions here. So we talked about how narcolepsy type 1 is caused by a loss of the orexin neurons, but the cause of NT2 is still unknown. Maybe some people have some partial loss of orexin neurons, but not really clear. The current meds are pretty good, but there's still improvements to be made on that. I do want to emphasize that, I'm talking about sort of broad strokes improvements here, but everybody's unique, and some people are going to do great with one drug and not so great with another. And this is why it's good to work with a doc who really understands the narcolepsy meds. And also, I just want to wrap up by talking about how drugs in development are potential treatments, but it takes years of work. I don't think it's a big secret for me to say that Takeda has been working on orexin agonists for almost 10 years. And they're working fast, and they're just starting to get into their phase 3 studies now. So it's a 10-year road to get these things from discovery to patients, and sometimes longer than that. And one thing I really want to mention is that studies are not possible unless people with narcolepsy and IH volunteer to be part of it. Because, and with so many drugs under development, there's a huge demand for people to join these studies. So I would really encourage you, because the more people that sign up for these clinical trials, the faster we're going to sort out whether, which are the best ones to advance into the clinic. Okay, so I'm going to stop with that and get to some of the questions. And I think what I'm going to do is I'm just going to do questions for about 10 minutes or so, if there's enough for that. Tammy, does that sound okay? Actually,
yes, there's a
there's not a lot in the queue.
Right.
Sorry.
There's a few open if you want to take those, and then I think we can close it down.
Sure. Okay. Yeah, and people feel free to type stuff, because I'm happy to stay a little over here. All right. So Beth is asking, with a recent NT2 diagnosis and no signs of cataplexy, would a sleep specialist help with the orexin levels? Is there somebody who could handle this? And so Beth, I would say there's a handful of places in the country that are sort of, well, actually, I shouldn't say that. I think it's certainly not unreasonable to discuss this with your sleep medicine doctor. But I think it depends. If you're in your first few years and you're worried about your orexin levels, it would not be completely unreasonable to do it. I have to say, for the most part, I think that it's just not worth the bother of doing a spinal tap in people with narcolepsy type 2, because almost all the time the orexin level is going to be normal and it doesn't really change anything. And you've gone through the headache of getting a lumbar puncture, but it's certainly something you could talk to your doctor about. I do not routinely recommend a spinal tap if it looks like somebody's got NT2. I do want to sort of leave that door open to what we talked about earlier, which is that possibly there could be some progression over the first few years if it's a new NT2 diagnosis. Okay, so Jennifer, this is more of a philosophical question, I think. Is it possible? So can we move away from the Epworth sleepiness score? It doesn't touch on most of my daughter's NT1 symptoms, like the chronic exhaustion, sleepiness, brain fog, no energy. It focuses too much on sudden sleepiness. Yeah, I hear you. The Epworth score is used because of just historical precedent and inertia. All sleep clinics know it. We continue to use it. It's got a lot of problems. One of my gripes about it is that sometimes people fill it out with the impression of like, well, do I feel like I could fall asleep watching TV? And I have to sort of emphasize, no, you're really supposed to answer, have you really fallen asleep? In the last week or two, have you truly fallen asleep? And so it's not a great tool, but it's kind of what we got right now. But yeah. Oh, actually, Jennifer, there is one other thing I'll say. There's something called the Narcolepsy Severity Scale, which was developed by Yves Dauvier, and it's really meant more for diagnosis and looking at treatment effects. But it's a great scale because it actually does get into those issues of cataplexy and cognition and things like that. And so it's not simply focused on just the sleepiness. Okay. Matt's asking, I've heard it's better to consolidate sleep and limit or eliminate naps. What would be considered a good or strategic use of naps versus using them poorly or over-relying? Oh, gosh. Well, I think everybody's a little different. But for most people, particularly with narcolepsy type 1 and for many with narcolepsy type 2, naps are great. And most people, if they can take a 15 or 20-minute nap and wake up feeling better for the next hour or two afterwards, that's terrific. And I really encourage people to try to integrate that into their work or school schedule. For IH, it's a different question. A lot of times with IH, people can take super long naps. If they fall asleep, they're going to sleep for 2 hours and then they feel junky afterwards and they don't want a nap. And so I would say in IH, we generally discourage naps. Outside of narcolepsy and IH, I do agree with what you say. It's good to consolidate sleep and eliminate naps. And so for people with insomnia, we often encourage them to not nap, but that doesn't really sort of apply so much to people with narcolepsy. Okay, Iris is asking about the keto diet. I don't have a lot to say on this. There's not been a lot of research on it. Some people would find that a keto diet is kind of energizing. I think trying it for a couple of weeks is fine, but there's all different kinds of stuff out there, and I'm just not expert enough to recommend anything in particular. I'd say this is something you probably want to, you might even want to start off by talking to your primary care doc, because a lot of sleep docs aren't going to be able to speak about that, and I sure can't. Marcia is asking, is it possible to end up with too much orexin from the orexin agonist meds? Will the body continue attacking it, or will replacing the orexin be enough where the meds could be stopped? Okay, there's a couple of things here. Is it possible to end up with too much orexin? Yeah, it's not actually orexin. The orexin agonists are just activating the receptor. So I think a better term might be, is it possible to end up with too much orexin tone or orexin signaling from too much of these meds? And I think the answer is definitely yes, because as you increase the dose in these clinical trials, you see more and more insomnia, more and more frequent urination, and that's not normal. And so it you know, there's one hopes there's a sweet spot somewhere where somebody is getting to a good level of alertness, but they're not having a whole lot of bothersome side effects. But I'm pretty sure that those side effects of insomnia and frequent urination are what we call on-target effects, which are due to excessive amounts of orexin signaling. Then the next part of her question is, will the body continue attacking it, or will replacing the orexin be enough where the med could be stopped? So the meds that are being developed are not physically at all like the orexin peptides. They're not like the native orexin molecules. So giving the meds should not kick up a hornet's nest. There's no evidence that it stirs up any kind of immune response. And the fact that the meds keep working for 8.5 months, as I showed you on that slide a couple back, is encouraging, that there's nothing sort of pushing back against the use of these drugs or producing some tolerance over time. Okay, so Amy Tesh is asking, do you know if most or any of these meds are including pediatric populations in their trials? My understanding is that Takeda is planning to do a pediatric trial once they're done with their adult studies. And I think it's a really important thing because, of course, narcolepsy begins in childhood oftentimes. And if anybody is in need of better meds, it's kids. And so I am eagerly looking forward to that. And I hope the other companies follow suit with that too. Chris is asking, do patients usually or always feel a cataplexy attack? Do you think patient-reported cataplexy attacks are underreported? So 2 interesting questions here. A lot of people, when I ask them about their cataplexy, says they can feel it coming on. And I've yet to find a patient who can actually put words to it. It's a weird internal feeling that we don't seem to have words for in any language. I don't know if Chris, you want to sort of type a little something to describe your feeling that an episode's coming on, but it's a lot of people say, I get a sense, I know it's happening, but it's really hard to put words to it. The second bit is, do you think patient-reported cataplexy attacks are underreported? And I think the answer is yes, because oftentimes when I'm seeing somebody in clinic and a parent or the spouse is sitting alongside them and I ask, so how much cataplexy are you having? And they're like, oh, maybe once or twice a week. And then the spouse shakes their head and it's pretty clear. It's like, no, it's like 5 or 6 times a week. Because I think when you have something all the time, and if it's kind of subtle, particularly just mild partial cataplexy, sometimes you just don't even notice. So I think that could be. But in terms of the clinical trials, if patients are reporting, if they're underreporting, well, then they're consistently underreporting. And so that's just sort of baked into the analysis. Kimberly is asking, do all meds become tolerable to the point where you need to up the dose to receive baseline response as when you started the med? And the answer there is no. In fact, I would say as a general rule, with the meds that we have now, and I hope for the future meds as well, most people, when we get them on a steady dose, do well on that dose for quite a long time. There's the exception to that is prior to, I don't know, 1990, sleep docs were giving sometimes really high doses of amphetamines. So in general, the max dose of an amphetamine type medicine is 60 or 70 milligrams per day. And docs back then were giving 120 milligrams, and it was so much that the body was truly becoming tolerant. But I think if the doses of any of these things are kept at a sort of a reasonable level, it's not common for people to become tolerant. If somebody says that they're doing fine on their meds and they're just cruising along for several years and then they start getting sleepy again, I go looking for other things. Maybe they have new sleep apnea. Maybe they started a different medicine that's causing some weird sedation. And so, but in general, the meds most of our meds hold up quite well over time. Ben is asking, my daughter has, I think that's narcolepsy type 1, and couldn't function on a placebo. Is there some way to participate in an orexin agonist study or get access before 2027? So the only way to get access is through the clinical trials. And with the clinical trials, you're kind of rolling the dice. Typically, they'll have 3 or 4 arms, meaning, 3 out of 4 people are getting the study drug and 1 group is getting the placebo. And that's unfortunately just how it goes. But it's necessary because you need to have a comparator, and I feel bad for the people on placebo. But typically after the 2-month study period, all the people who were on placebo are offered active drug. And so then you can go into the open label extension and get access to the drug that way. So I think that's pretty much the only option for now.